Hello guys, today we shall be discussing another important topic in microbiology, that is your measles virus. Okay, that is your measles, which is also called as rubeola. So regarding the measles virus, what all things we get to know? So first of all, let me write down the heading. Measles. So what is the other name for measles? Measles is also called as rubeola. Measles is also called as rubeola. Now don't confuse this with rubella, because rubella is different thing. Rubella is called German measles and this is normal measles, okay, which is also called as rubeola. Now if you look at the family, genus and the species, which you really need to know in your microbiology, right? So regarding the family, regarding the genus and regarding the species. Now regarding the family, what is that important thing you need to know? This measles or rubeola, it belongs to a family called as Paramyxoviridae, right? It belongs to a family called Paramyxoviridae. I hope all of you are taking the notes. And what is the genus here? It belongs to a genus called as Morbillivirus. Morbillivirus. Bah, super. It belongs to a genus called as Morbillivirus. Morbillivirus, right? Morbillivirus. And what is the species? The species is your measles virus. Species is your measles virus. So these are the three important things which you need to know. Now apart from these things, first very important thing is that how this measles virus is gonna get transmitted from one individual to another individual, right? From one child to another child. Mostly the school age group, right? Because they all of them sit in one classroom, that if one child is affected with this measles, through the respiratory droplets, I mean through airborne contact. Okay, so through the respiratory droplets, if one talks loudly or one sneezes, one coughs, what will happen is that through these respiratory droplets, the virus gonna land upon the other individual. Okay, so if I talk about the transmission, if I talk about how this virus is transmitted. So what is the mode of transmission of this virus? The mode of transmission of this virus is pretty simple, and that is your what? That is your respiratory droplets. That is your respiratory droplets. Now apart from this respiratory droplets, what is the other thing you can tell? The risk factor. Which group of people are under high risk? So every virus, the first group of people which will be under higher risk are those who are not vaccinated. Basically, you have got a vaccine called MMR vaccine, measles, mumps, rubella vaccine, right? So if you have taken this vaccine in your childhood, then you are less likely to get affected with this infection. But if you have not taken any kind of vaccination, then you are more likely to have this infection. So if I just jot down the points on the risk factors, jot down the points on the risk factors. What are the risk factors which you can see in this patient over here? The first important risk factor is, what is the first important risk factor? That is unvaccinated individuals. Unvaccinated individuals. Apart from this unvaccinated individuals, those who are staying in an endemic areas. So unvaccinated individuals as well as those who are staying in the endemic areas, both these groups are under high risk of getting this measles virus. So as I told you, measles virus, it mainly transmits through respiratory droplets. So how this virus is going to enter into your respiratory system? And in the respiratory system, what is happening to this virus, where this virus is replicating, right? And after replication, all these virus particles, how they are spreading to different parts of the body and how they are infecting you, we shall be discussing right now. Okay. So if you look at the virus, guys, this is just a rough picture of the virus. So this is an enveloped virus, by the way. It has got a double membrane over here, right? Now if you look at the virus, there are few important things. For example, you have got such few important things on either side like this. Now what are these important things which you find on the either sides? These are called as F protein. What is F protein? F protein is also called as fusion protein. F protein is called as fusion protein. What is the function? I'll let you know. Apart from that, you have also got some more interesting structures, right? Some more interesting structures like this. Now what is this interesting structure which you can find? Now this is found all over. Obviously such proteins are found all over. So what is this interesting structure, guys? This is called as H protein, or also simply called as hemagglutinin protein. So what is the name of this? Yes, the name of this is H protein. Okay, so what will be the function? This is also called as hemagglutinin protein. So if you look at the surface of this measles virus, you're going to find two important particles. One is called as the F protein, or called as fusion protein. Another one is called as the H protein or hemagglutinin protein. Now let us dive into the pathogenesis. Now please don't write any notes, just listen to the pathogenesis which I am talking about. Okay. So let me draw a picture before I teach you the pathogenesis. Okay. So if you can look here, that as this is spreading through respiratory droplets, it is very important that I draw the lateral surface of the man's face. Right? Now all of you are very clear in this concept that you have got a beautiful oropharynx over here. You have got a very beautiful nasopharynx over here. Your nasopharynx and oropharynx, anyways I'm not going into anatomy, your nasopharynx and oropharynx together they join and form this structure called as laryngopharynx. So if I make a small division in it like this, the anterior part will become your trachea, the bronchi and the lungs. The posterior part goes down all the way and ends up having your stomach, right? So this is how it continues like this. Right? So this is how it's gonna continue, and obviously you gonna have your intestine all the way like this. This is your intestine. Anyways, I'm not going into that deeper. Next important thing is that with this pink color thing I'm drawing here, that this is all the trachea. This is all the trachea which divides like this, and here comes your beautiful lungs. Clear? Now there is no fun in telling that you have got an important part which we use rarely, that is the brain. Okay, and there is another important thing that I want to draw here, that is your blood vessel. So this is the representation of all your blood stream, right? So the entire blood vessel which you have drawn, each represents your blood stream. So what I'm talking about is that if you can look here, guys, very important thing I'm telling you, that through the respiratory droplets the virus particle enters inside, right? So look here. This is the virus particle. Let me keep the virus particle red. Okay, so this is the red color virus particle. Now this red color virus particle is right now in your nasopharynx. Now from the nasopharynx it is descending down into your lungs over here, right? Now where is this virus particle? Exactly in the bronchi here, exactly in the bronchi. So if I dilate the bronchi, or if I show you this entire picture little more bigger, right, how does this picture look is that, look here guys, it's very important. So these are, let us say, so these are your dilated bronchi here. This is your dilated bronchi, right? And obviously what was this? this is your lung tissue. That is your bronchi and this is your lung tissue. So you know that this part is your lung tissue. Okay. So, and let me also draw an alveoli here, right? So this is how that was represented here. Now look here. Obviously, all of you know that lining we have got bronchial epithelial cells, right? So we have got a bronchial epithelial cell here, we have got the same cell on the opposite side, we have got another cell over here, the same on the opposite side like this. But what I'm doing is I'm just enlarging one cell like this. I'm just enlarging one bronchial cell. Now where is this virus particle? Where is the virus particle? You see, this is the virus particle over here. Right? So right now the virus particle, as you can relate with the previous picture, the virus particle is right now here. Now this virus particle, guys, this virus particle, if you very closely look, this virus particle, I told you it has got two important parts, two things which are coming like this. These are called as fusion proteins. So where are the fusion proteins here? Look here. There is one fusion protein here. There is another fusion protein here. There are two fusion proteins. What is the use of these fusion proteins? These fusion proteins fuse this virus particle to the cell. Let us say that my hand is a virus particle. Now on the surface of my hand you find a long big protein. This is called fusion protein. With the help of this fusion protein, this is my host cell. Now with the help of this fusion protein, this virus particle is having this fusion protein and this fusion protein is fusing into the host cell. Or in other words, I can tell this fusion protein is fusing this virus particle to the host cell completely. It is making the complete attachment to the host cell. On the other hand, I also told you there is another protein that is called as hemagglutinin protein. So before I discuss that, you should know that on the bronchial respiratory epithelial cell, right? I'm not discussing what type of epithelium you have here. So this is not histology anyways. So on the bronchial respiratory epithelium, you have got a special type of receptor. Now if you can see here very clearly, this receptor which I'm drawing like this right now here, this is called as CD46 receptor, cluster differentiation 46 receptor. And mind you, this receptor is present on every cell which has got nucleus in it. So you know in your body how many cells have got nuclei in it. Every cell has got nuclei in it. It means every cell is having this receptor, and this receptor, my friends, is called as CD46 receptor. Now this virus particle also has got, I told you, a very special structure that is coming out. What was that? Hemagglutinin. Now look here, this hemagglutinin fuses with that CD46. After fusing, right, after attaching, it attaches to that receptor, and these fusion proteins, so they completely bring the virus particle down and fuse with the membrane. So as a result, what is happening? As a result, what is happening is that the virus particle now enters into your respiratory epithelial cell. Right? So right now, guys, this is the virus particle. This is the virus particle right now into your respiratory epithelial cell. And one very important thing that I should mention you before, remember that thing, that this virus, if you look at the genetic material of the virus, look here, this virus is a single-stranded RNA molecule. Okay, so what is this virus? This virus is a single-stranded RNA molecule. Single-stranded RNA molecule. So I basically call this as negative sense single-stranded RNA molecule. Negative sense single-stranded RNA molecule. Can you please explain what is this negative sense single-stranded RNA molecule? There are two types of RNA molecules, guys. One is called positive strand. Another one is called as negative strand. I mean positive sense, negative sense. Positive, listen very carefully. Positive sense RNA molecule. What is the basic function of RNA? Tell me. RNA molecule, right? What is the basic function of RNA molecule? For example, let us say this is an RNA molecule. What is the function of that? RNA molecule will undergo transcription or translation. Obviously, it will undergo translation and synthesize what? Proteins. There are some RNA molecules there are some RNA molecules. Now these RNA molecules have to be processed. Processed in the sense, first this RNA molecule will convert into another RNA molecule, and this RNA molecule will finally synthesize proteins. Now what is the difference between the top one and the bottom one? The top one is an RNA molecule that is directly translating into proteins. So that is called as positive sense single stranded RNA molecule. But the bottom one here, this is a negative sense single stranded RNA molecule. So this is negative sense single stranded RNA molecule. What is happening to the negative sense single stranded RNA molecule? With the help of an enzyme, RNA polymerase. With the help of an enzyme RNA polymerase, negative sense single stranded RNA molecule is converting into positive sense single stranded RNA molecule, and this positive sense single stranded RNA molecule will finally end up into proteins. So this is a simple difference between what is negative sense, what is positive sense. This measles virus, guys, this is a negative sense single stranded RNA. So what it should happen? What should happen for that? Look here very carefully. So this is the RNA inside. This RNA is coming out now. Right? Now this RNA should first be converted into positive strand RNA molecule. So it is getting converted into positive sense single stranded RNA molecule. Now this positive sense single stranded RNA molecule will now synthesize what? It will synthesize proteins. So it is now synthesizing its molecules like this. These protein molecules, or simply called as the viral particles. Right? Now these viral particles, what will happen is that, look here very carefully now, these viral particles will enter into the interstitium of the lung. How are they going to enter? Before entering, it has to cross that lipid membrane, right? So this pink color line which I have drawn down here, this is the lipid membrane. So it has to cross that lipid membrane and then enter. Look here now. So what I'm trying to tell you is that this is the virus particle. Now it has to cross this lipid membrane, right? So what it will do, this virus particle, as it is going outside, it will take up that lipid membrane, bind around itself, and then enter into the interstitium. Look here how it is entering. So finally the virus particle has entered into the interstitium and it has taken up the lipid membrane surrounding that. Now you understood? So in this way, many virus particles are over here, many of them. In this way, many virus particles are here, like this. Now, when this virus particle, before I discuss this thing, look here now. Question number 1, where did the replication of virus particle happen so far? The replication of virus particle happened in the respiratory epithelium. That is a point to be noted. Point to be noted is that virus replication, where did it happen? In the respiratory epithelium. Virus replication happened in the respiratory epithelium. Okay, not only respiratory epithelium, save your space here. I am also going to tell another important part where the virus replication will happen. Now look here, virus replication has happened finally from the respiratory epithelium. The virus have taken the bronchus membrane, the lipid membrane, and finally entered into the interstitium. Now within the interstitium, you have got two important cells. One are called as alveolar macrophages and the other cell is called as fibroblasts, right? One of the two important cells, look here, these cells which I am drawing here, these cells are your alveolar macrophages. Right? And the other group of cells which I am not intended to draw here, those are called as fibroblasts. Why I am not drawing? Because both of them have the same function. What these alveolar macrophages do is that these alveolar macrophages will take up that virus particle in this way. These alveolar macrophages are taking up that virus particle. Even the fibroblasts do the same thing. So alveolar macrophages are taking up that virus particle, and you know, virus particle is so intelligent that this measles virus even starts dividing within the alveolar macrophages and also starts dividing within the fibroblast cells. So if I add a second point, that virus replication is happening at two places. One is your respiratory epithelium and the other one is your alveolar macrophages and fibroblasts. Alveolar macrophages and your fibroblasts. So these are the two places where your virus replication is happening. Now once virus has been replicated, what is the next step? What is the next step is that right now within your alveolar macrophages you have got very good quantity, right? You have got very good quantity of these virus particles within your alveolar macrophages and fibroblasts. Now these alveolar macrophages or fibroblasts, they travel all the way because they are motile, they travel all the way, finally enter into the lymph nodes, right? Now you might get a doubt, you did not draw any lymph node here. Yes, I will draw a lymph node now. Now look here, these are the lymph nodes. You know, everywhere in your body you have got the lymph nodes. So these are the lymph nodes like this. So all along your lung over here you have got these lymph nodes. So where is the virus particle now? Right? Tell me guys, look at this picture. I'm zooming this picture. Where is the virus particle? This is your alveolar macrophage, right? So this is the virus particle, and surrounding your virus particle you have got the lipid membrane like this, right? So this is the virus particle right now here. Now this virus particle, now very nicely it enters into your what? Lymph nodes. Now within your lymph nodes also I am putting a dot which represents the virus particle even in the lymph nodes also. So the first place from the lungs where the virus particle goes is to your lymph nodes. So when the virus particle is going to your lymph nodes, don't you think patient will have lymphadenopathy? Yes, obviously the patient is going to end up having a condition that is called as lymphadenopathy, right? So this is the first important thing. That is when a patients of measles or mumps or rubella or whatever it is, the lymph nodes are first thing which are affected, right? So that is the reason why they have lymphadenopathy, swollen lymph nodes. Even in measles patient, if you try to palpate the lymph nodes, you will find swollen lymph nodes, right? So lymphadenopathy is the first important thing. Apart from this, the virus, right, the first infection started in the lungs. Obviously, the virus replicated in the lungs, entered into the interstitium, and macrophages and fibroblasts have taken it up, and within the macrophages and within the fibroblast cells, the virus replicated again, right? So obviously, there will be a quite very good infection that is happening within the lung interstitium. And what is that infection? That infection is nothing but pneumonia. So obviously within the patient's lungs also you're gonna find an infection, and that is named as pneumonia. So you're gonna see pneumonia in the patient as well. Not only that, even now this virus affected your lungs, you know how it affected. It affected your lymph nodes, you know how it affected. Now this virus, even guys, look here now, very interesting. This virus from here it is even gonna enter into your stomach. From the stomach this virus gonna reach your intestines like this. You see, this virus is reaching your intestines. Now don't confuse, this is the virus which I have drawn before. So this is a pink lipid membrane surrounding it. It is reaching your intestine, and when the virus reaches your intestine, don't you think it is gonna damage your intestine? I mean, virus, see guys, any disease, any pathology, the bacteria directly does not damage your body. The virus directly does not damage your body. When there is a virus there, this virus will attract all your immune cells. Looking at that virus, all your immune cells will come there. So immune cells don't know which place they are fighting, nothing like that. If it sees the virus, even if it is on the heart, even if it is on the lung, even if it is on the brain, it goes and fights with that. So in the process of fighting, who is affected? The underlying tissue is affected. right? So if the fight is happening in the intestines, the intestine is affected, and this results in the patients getting diarrhea. So patient will also have a manifestation of diarrhea, right? Diarrhea. Apart from this, another important thing is that this virus now very neatly it enters into your bloodstream. This virus is right now within your bloodstream. Okay, now within the bloodstream, what will happen to this virus is that this virus will ascend up all the way, and finally this virus is going to enter into your brain, right? So this virus will enter into your brain. Now once this virus is in your brain, this is going to cause a really bad thing which is not at all glamorous. So that is your encephalitis. So patient is also going to have another condition, that is encephalitis. That is encephalitis, right? So can I write down all the points, guys? Can I write down all the points, and let us discuss all of these things, whatever things we have discussed so far, right? First thing, the infection was in the lungs causing pneumonia. From the lungs, the alveolar macrophages have taken up that infection, the fibroblasts have taken up that infection, went to the lymph nodes, given to the lymph nodes. Patient is having lymphadenopathy. From the lymph nodes, it went to the intestine and caused diarrhea. And from there, the infection, the virus entered into your bloodstream, it ascended up all the way, entered into your brain and caused encephalitis. Encephalitis. So all these are the symptoms which you're gonna find in the patient. All these are the important symptoms which you're gonna find in the patient. The major important clinical manifestations which you'll find. Apart from this, can you also tell another thing which can be found in the patient? Yes, obviously. Another important thing is that, guys, look here very carefully now. Look here very carefully. Let us say this is the line which I am drawing for the disease starting to the ending point. Okay. So let us tell that I got the infection today. When did I get the infection? I am infected today. I am infected today in the sense the virus entered today. Infected or virus entry. Virus entry happened on day 0. Right? On day 0 the virus entry happened. Now, if virus entered into my body today, tomorrow I will be today I will be fine. Tomorrow I will have no symptoms. Day after tomorrow also I will not have symptoms. For 5 days or 6 days I won't have symptoms. It means for how many days you are not having symptoms? Okay, you will not have any symptoms for about 10 to 14 days. For 10 to 14 days you won't have any kind of symptoms. For 10 to 14 days you look so fine. Now this stage of fineness, even after the virus entry into your body, till 2 weeks, you are not having any symptoms, then that phase is called as incubation period. And that is called as your incubation period. During this incubation period, virus is preparing inside slowly. It is slowly dividing. It is entering into macrophages. It is entering into fibroblasts, slowly dividing, right? Now, after 14 days, the virus particles are ready. Right, the entire military is ready. There are thousands and thousands of virus particles right now waiting to just cause infection in your body. After 14 days, right, after 14 days, in the next 3 days, what is happening is 14 days in the sense, in the next 3 days in the sense 15, 16, 17 or 18, let us say. So these 3-4 days, right? So these 3-4 days, what will happen is that the virus suddenly explodes like this. I mean, the virus particles, they suddenly enter into your bloodstream and cause havoc things, right? So these virus particles are entering into the bloodstream and it will cause very high fever. Suddenly you're gonna have high fever overnight. So that phase you call it as prodromal phase. That phase you call it as prodromal phase, right? So this is your prodromal phase. In your prodromal phase, you're gonna find three important symptoms. CCC. What is this CCC? CCC stands for cough, coryza and conjunctivitis. You're gonna find cough, coryza, conjunctivitis. So these are the three C's which you're gonna see in a patient in the third day, like that. So after having cough, coryza, conjunctivitis for 3 days, continuously for 3 days, on the next day, right, after the fourth day or the fifth day, the patient will have another important problem. That patient will slowly develop some very special rashes inside the mouth. So those special rashes in the mouth are called as enanthema. So after 3 days of severe fever, right, after 3 days of high fever with cough, coryza and conjunctivitis, right, the highly infectious phase, after this then patient is going to have some very important rashes within the mouth, important spots within the mouth. So in the next few days the patient is going to have what? Those important spots within the mouth, and they are called as Koplik spots. Right, they are called as Koplik spots. And again I'm telling you, Koplik spots are pathognomic for this measles. In almost all the patients of measles, you're gonna find Koplik spots, right? So these are the spots which are present where? In your mucous membrane, below the tongue, on the tongue, inside the buccal mucosa, you will find these Koplik spots, right? Okay, fine. So after Koplik spots, okay, so you told me that virus entered today, for 10 to 14 days I have no symptoms, then suddenly continuously for 3 days I have very severe symptoms, right? And after 3 days of very severe symptoms, then suddenly I find some spots developing in my mouth. Okay, fine. So after that, after developing these Koplik spots, the next day itself you will even find the spots and rashes developing on your body, right? So in the next 4 days, you will find the rashes that are developing on your body, right? Now what are these rashes that are developing on your body? These are called as exanthem. What are these called? These are called exanthem. Now what is your concept of exanthem, guys? Exanthem means these are maculopapular rashes that start from the head, they come down all the way to your extremities. It means first they affect the trunk, then they go to the extremities. So center to periphery. Such type of maculopapular rash is called as centrifugal rash. So point number 1, it is what type of rash? It is maculopapular rash. How it is progressing? From the head to the toe, to the extremities, from top to bottom, that is cephalic to the caudal, cephalocaudal progression. Cephalocaudal progression. And third important thing, what type of distribution is this? Centripetal or centrifugal? Centrifugal, from the center to periphery. Fine. The remaining period, after getting rash, I will have high fever. After that, the next day I will have spots in my mouth, right? After that, for 4 days I will have rashes, and after that slowly the symptoms will resolve. Slowly the fever will be reduced, slowly the Koplik spots will be reduced, slowly these exanthem, they will disappear slowly. I mean the maculopapular rashes slowly go. So within the next 10 to 14 days, I am slowly recovering from this problem. That is why I call this phase as a recovery phase. Right? So this is the very important part, guys, which I would want you to remember. This is a very important part. Okay, so keep these things in mind. Clear? So many MCQs are framed from this part especially. The next important thing is, guys, what are the complications? What complications would I see if I am not treating this patient properly? The complications which I would see is that obviously patient's lungs are affected, so there can be gained cell pneumonia. Just remember it. Right? Apart from this gained cell pneumonia, patient will also have subacute sclerosing panencephalitis. It means there will be complete sclerosis of your brain tissue. So that is called as subacute sclerosing. Pan means total. Sclerosing panencephalitis means total brain is getting infected. Total brain is undergoing sclerosis. Panencephalitis. So SSPE, subacute sclerosing panencephalitis. So in this, what is happening is the entire brain is getting infected. Actually, this subacute sclerosing panencephalitis is also called as a neurodegenerative disease. So when you call it as a neurodegenerative disease, when you call this as a neurodegenerative disease, what do you mean by neurodegenerative disease? It means that all the neurons are degenerating. When all the neurons are degenerating, there are neurons from the brain which come down to your muscles. These are called as motor neurons. So when the motor neurons are getting degenerated, will the patient have normal motor function or motor dysfunction? Obviously, because of this neurodegeneration, patient will have motor dysfunction. So when patient will have motor dysfunction, the reflexes will not be proper. Either there can be hyporeflexia or hyperreflexia. When there is any problem in the brain speaks only one language, that is seizure language. So any problem with the brain, patient will have seizures, right? If patient is having seizures, there is a problem with the brain. You need to rule out what is that. Maybe because of fever, maybe because of let us say high blood pressure, maybe because of some bacteria, some virus. Everything can cause seizures, right? So motor dysfunction will happen in the patient. So in these patients, guys, in subacute sclerosing panencephalitis, when I take an MRI of the brain, some places I will find very hyper intense lesions. I will show you the MRI shortly, but just believe me that you will find very whitish lesions. These whitish lesions are called as hyper intense lesions. After the complications, how do I diagnose this case? I diagnose by what is the lab tests which I do. The first important lab test which comes to my mind is that what if I do a lymph node biopsy? Lymph node biopsy. Look here now very carefully. This is your trachea, this is your bronchi, right? So as I already spoke this thing that you have got lymph nodes like this. I am just enlarging them. You have got lymph node network like this. Virus particle is right now within your lymph node, right? And within your lymph node you have got what cells? Lymphocytes, right? Within your lymph node you have got lymphocytes. I am enlarging one lymph node now. Look here, this is one lymph node. Now within this lymph node you have got the virus particle like this, and obviously there is no fun that you have got a lipid membrane surrounding that. So within the lymph nodes you have got lymphocytes. So these are the lymphocytes now. All these are your what? All these are your lymphocytes. Obviously lymphocytes will try to attack the virus because the virus has entered into its home, that is lymph node. Now these lymphocytes will try to attack that. Now what will happen, the virus is so tricky that a single lymph node cannot go and fight. So what will happen is that when you unite with everyone, you become stronger, right? So all the lymph nodes will unite with one another. When all the lymph nodes will unite with one another, don't you think the entire unit will be so big? Yeah, so this is how the entire unit looks, and when all the lymph nodes unite together, this is how the nuclei are. All the nuclei are at one place. And now this lymph nodes, all of them, they join together to form a single giant cell, right? So this is also called as giant cell. Giant cell. Other name for this is Warthy-Finkeldy cell. Not Warth, it is Warthin. Warthin Finkeldy cells. Right? So from today onwards, if someone asks you what are Warthin-Finkeldy cells, they are nothing but multinucleated gain cells. Right? So what are they? They are simply multinucleated gain cells where all the lymphocytes within the lymph node join together to form this Warthy-Finkeldy cells. So this is the lab diagnosis which I do. Apart from this, I also check for the measles antibodies, either IgM or IgG. And what will be the treatment, guys? What will be the treatment in these patients? There is no specific treatment. The treatment is mainly supportive treatment. In this supportive treatment, whenever patient is having any kind of infection, the first thing that happens to the body is dehydration. So you need to put NS to the patient, I mean normal saline, run the fluids to the patient and hydrate the patient. Second thing, patient is in pain, you need to give analgesics. And third thing, patient is having high fever. When? During the prodromal phase. So you need to give antipyretics to the patient. So this is the basic treatment part which you need to know. So this is all the thing you need to know regarding the measles virus. Let me put up the pictures. This is the first picture which I would like to show you. You see in the mouth, you see these red color spots over here, right? So these spots are called as Koplik spots. These are called as Koplik spots. Koplik spots are characteristic for measles. The second important thing, this particular rash which you can see here, this is called as maculopapular rash, right? And third important thing, one of the complication of measles which I told you, subacute sclerosing panencephalitis, where you will find hyperintense lesions, right? You can see the lesions here on four corners here. So these are very white in color, right? So these are hyperintense. So such lesions are called as hyperintense lesions. So patient is having hyperintense lesions, that is SSP, right? So this is all you need to know about the measles. So thank you so much for watching my video. Goodbye.